Hello everyone, my name is Sarah, and this is the NCERT audiobook series for you all. Our today's topic is Plant Kingdom, Part 3, and we will cover today the Pteridophytes and Gymnosperms. Before we start the audiobook, must press the like button and subscribe yours beautiful channel Eduholic, official. So, let's get started.
So, let's begin the audiobook with Pteridophytes. The Pteridophytes include horsetails and ferns. Pteridophytes are used for medicinal purposes and as soil binders. They are also frequently grown as ornamentals. Evolutionarily, they are the first terrestrial plants to possess vascular tissues, xylem and phloem. The Pteridophytes are found in cool, damp, shady places, though some may flourish well in sandy soil conditions. You may recall that in bryophytes the dominant phase in the life cycle is the gametophytic plant body. However, in Pteridophytes, the main plant body is a sporophyte, which is differentiated into true root, stem and leaves. These organs possess well-differentiated vascular tissues. The leaves in Pteridophyta are small, microphyls, as in Selaginella, or large, macrophyls, as in ferns. The sporophytes bear sporangia that are subtended by leaf-like appendages called sporophylls. In some cases, sporophylls may form distinct compact structures called strobili or cones, Selaginella, equisetum. The sporangia produce spores by meiosis in spore mother cells. The spores germinate to give rise to inconspicuous, small but multicellular, free-living, mostly photosynthetic thalloid gametophytes called prothallus. These gametophytes require cool, damp, shady places to grow. Because of this specific restricted requirement and the need for water for fertilization, the spread of living Pteridophytes is limited and restricted to narrow geographical regions. The gametophytes bear male and female sex organs called antheridia and archegonia, respectively. Water is required for transfer of antherozoids, the male gametes released from the antheridia, to the mouth of archegonium. Fusion of male gamete with the egg present in the archegonium result in the formation of zygote. Zygote thereafter produces a multicellular well-differentiated sporophyte, which is the dominant phase of the Pteridophytes. In majority of the Pteridophytes, all the spores are of similar kinds. Such plants are called homosporous genera like Selaginella and Solvinia, which produce two kinds of spores. Macro, large, and micro, small, spores are known as heterosporous. The megaspores and microspores germinate and give rise to female and male gametophytes, respectively. The female gametophytes in these plants are retained on the parent sporophytes for variable periods. The development of the zygotes into young embryos take place within the female gametophytes. This event is a precursor to the seed habit, considered an important step in evolution. The Pteridophytes are further classified into four classes: Psilopsida, Xylotum, Lycopsida, Selaginella, Lycopodium, Sphenopsida, equisetum, and Pteropsida, Triopteris, Pteris, Adiantum. Now, our next topic is Gymnosperms. The Gymnosperms, gymnos means naked, sperma means seeds, are plants in which the ovules are not enclosed by any ovary wall and remain exposed, both before and after fertilization. The seeds that develop post-fertilization are not covered, i.e., are naked. Gymnosperms include medium-sized trees or tall trees and shrubs. One of the gymnosperms, the giant redwood tree sequoia, is one of the tallest tree species. The roots are generally tap roots. Roots in some genera have fungal association in the form of mycorrhiza, pinus, while in some others, cycas, small specialized roots called coralloid roots, are associated with N2, fixing cyanobacteria. The stems are unbranched, cycas, or branched, pinus, cedrus. The leaves may be simple or compound. In cycas, the pinnate leaves persist for a few years. The leaves in gymnosperms are well adapted to withstand extremes of temperature, humidity, and wind. In conifers, the needle-like leaves reduce the surface area. Their thick cuticle and sunken stomata also help to reduce water loss. The gymnosperms are heterosporous, they produce haploid microspores and megaspores. The two kinds of spores are produced within sporangia that are borne on sporophyls which are arranged spirally along an axis to form lax or compact strobili or cones. The strobili bearing microsporophyls and microsporangia are called microsporangiate or male strobili. The microspores develop into a male gametophytic generation which is highly reduced and is confined to only a limited number of cells. This reduced gametophyte is called a pollen grain. The development of pollen grains take place within the microsporangia. The cones bearing megasporophyls with ovules or megasporangia are called macrosporangiate or female strobili. The male or female cones or strobili may be borne on the same tree, Pinus. However, in cycas male cones and megasporophyls are borne on different trees. The megaspore mother cell is differentiated from one of the cells of the nucellus. The nucellus is protected by envelopes and the composite structure is called an ovule. The ovules are borne on megasporophyls which may be clustered to form the female cones. The megaspore mother cell divides meiotically to form 4 megaspores. One of the megaspores enclosed within the megasporangium develops into a multicellular female gametophyte that bears 2 or more archegonia or female sex organs. The multicellular female gametophyte is also retained within megasporangium. Unlike bryophytes and pteridophytes, in gymnosperms the male and the female gametophytes do not have an independent free-living existence. They remain within the sporangia retained on the sporophytes. The pollen grain is released from the microsporangium. They are carried in air currents and come in contact with the opening of the ovules borne on megasporophyls. The pollen tube carrying the male gametes grows towards archegonia in the ovules and discharge their contents near the mouth of the archegonia. Following fertilization, zygote develops into an embryo and the ovules into seeds. These seeds are not covered.
Thanks for watching the complete video. Don't forget to watch the previous parts of this series, link is given in description. See you all in the next part. Till then, stay safe, stay focused.